Well, good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Biomonitoring, Relevance of Bioassays in Water Treatment. My name is Samaya, and it's my pleasure to be your X Talks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved, so please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, you can contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please also note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Indigo Biosciences who developed the content for this presentation. Indigo Biosciences is a leading provider of nuclear receptor and in vitro toxicology solutions that accelerate scientific decision-making. They supplement the world's largest portfolio of nuclear receptor kits and services and in vitro toxicology solutions with greater results, readability, reproducibility, and faster turnaround times. The solutions, plus supportive team and reliable science and platforms, aim to reduce the time, cost, and risk associated with the discovery process. Now it's my pleasure to introduce our speaker for today's event, and that's Dr. Jack Vanden Heuvel. Dr. Jack Vanden Heuvel is a recognized expert in the field of nuclear receptor biology and toxicology with over 100 peer-reviewed publications. In addition to his role as CSO at Indigo, Dr. Vanden Heuvel is a professor at Penn State University, where he is program coordinator of the undergraduate toxicology program, co-director of the Center of Excellence in Nutrigenomics, and leads an extramurally funded research program. So now, without further ado, I'd like to hand the mic over to our speaker. Jack, you may begin when ready.
Well, thank you, Samaya. And it's a pleasure to speak with you today about what's an exciting and growing area, not only at Indigo Biosciences, but within my academic lab, and certainly within a lot of government agencies, both nationally and internationally. And that's using bioassays, cell-based assays, to look at water quality and the results of water treatment and recycling programs. My talk is going to be at a relatively general level without a great deal of details about chemistry or molecular biology. What I'd like to do is kind of give you enough information to appreciate the field and what direction it's going in. So I'm going to first talk about the problem. What are the current state of looking at water quality, and then start to go into a general approach of how you incorporate some cell-based assays into looking at water quality, talk about how you pick the bioassays that are going to give you the most useful information about water quality, and then give some examples and wrap up with some future directions and conclusions. So as many of you know, the testing of water quality has been going on for a long time, and it really has a very standardized set of parameters that are being looked at. So generally speaking, water samples are collected, and then a variety of physiochemical parameters are examined. Things like total organic carbon, the pH, turbidity, color, nutrients, minerals, etc. And all of these different endpoints are examined and then compared to what is permitted. What are the maximum permitted concentrations? And that's what's used to make decisions about water quality. Where we run into problems is the man-made chemicals that are often present in a variety of different water, and how we go about making decisions based on those micropollutants. And generally speaking, our current approach is to kind of look at priority substances that are in this pool of chemicals within the water, and look at the concentrations of those selected compounds using very typical analytical chemistry approaches. And we do this for a lot of different chemicals, including metals and pesticides. And then we regulate based on the concentration of these priority chemicals. Where we run into problems is with those chemicals that are present at low levels and chemicals that we don't have a lot of information on. And these collectively are called contaminants of emerging concern. And this is a general term that is used to talk about the broad class, but this is really many different types of chemicals that are coming from multiple sources. And they get this term emerging because they may be newly manufactured, they may have just been identified, they could be naturally occurring chemicals that haven't been scrutinized through the regulatory agencies. So in most cases, these contaminants of emerging concern have not been looked at from a regulatory standpoint. But that's not to say that they are without consequence, because many of these compounds are suspected to have ecological and human health effects. And in addition, many of these compounds or contaminants of emerging concern are consistently found in different sources of water. So we can find them in groundwater, surface water, wastewater treatment facilities, soil, food, etc. And many of the problems of these compounds are not only their presence and the fact that we don't know much about them, but the fact that they accumulate. So they stick around longer, they're harder for us to handle as organisms, to try to metabolize and get rid of. They accumulate in crops, so therefore they're going to be accumulating in us. They may accumulate in the food chain. So we are developing a sense that some of these compounds are persistent. And we really don't have a good handle on how to test for these different compounds. So we need better ways of looking at these compounds that we have little knowledge of. We don't know what sorts of health effects they have, which sorts of biological and environmental health effects that they have. And certainly some of these compounds are present at very low levels, which doesn't mean that they don't have detectable toxicity. So we need a way to detect these low concentrations of these unknown chemicals. So right now, the way that we handle these compounds of contaminants of emerging concern is analytical chemistry. So as I mentioned before, we may pick some target chemicals which have some known effects or fit into a known class of compound, design analytical tools to be able to extract those and to quantify their presence in particular matrices. And then model that to see how much we may be exposed to in water, model how much would be expected to end up in different biota within the environment. And for many compounds, we can use this approach and get a good handle on the overall quality of that particular water sample. Where we run into problems is for those compounds that we do not know if they're present, or if we don't know what their degradation or persistence is in the environment. And the other overriding factor when you start to think about these complex mixtures of chemicals is that they are present in very complicated mixtures and complicated matrices that may change over time. So, of course, very challenging to start looking at all of these different unknowns coming from a variety of different sources. And the challenges are are plenty, and I'm showing three major challenges here. So these chemicals, even though we can detect them, we can measure them, we may not know what their toxicity is. These fit that definition of an emerging contaminant, where we do not have background knowledge on the types of toxicity or at what doses these chemicals cause a toxic response. We may have difficulty in the methods that are required to detect certain unknowns. If we don't know they're there, we don't know how to develop analytical tools to measure them. And then finally, something that I suggested before is the fact that these are present as complex mixtures. And so we have to come to grips with how these chemicals are interacting with each other, how they might be able to have additive effects, synergistic effects, or more than additive, or one chemical may be interfering or antagonizing the effect of another chemical. So this complexity and detection limits, all of these things add together into the fact that we need better tools to assess the quality of different water samples. And what we're looking for is a way to look at the sum effect of all of these chemicals that are present at a given time in a given water source. And what I'm going to be talking about is one potential avenue that we can explore to look at that cumulative effect. And that's using bioassays. In particular, bioassays that are looking at particular mechanisms or effects that we can use. So here's the general approach that we're going to describe, that is starting with looking at different water samples and hopefully at the end of the process being able to make some decisions about the quality of that water or the effect that a treatment has had on the quality of that water. And I've divided this up into four major steps that are required to use these bioanalytical tools. The first is sample collection. Second is sample extraction. The third would be picking the toxicologic or pharmacological endpoint that you want to examine. And then the fourth is trying to match a bioassay for that endpoint that you have selected to interrogate. And we are going to spend the vast majority of this talk on point number 4, but I thought I would spend this slide at least talking about the first 3 steps in this process, just so that you can put the bioassays into context. So the first, of course, is sample collection. And this particular talk is dealing with water quality. And of course, these assays can be used for a variety of different types of water. They're quite frequently used for looking at wastewater treatment facilities, recycling programs. These could be environmental samples, surface water, groundwater. These could be from sediment samples, sediment water, or it could be any step within the water recycling and purification process. These can also be from other biological matrices as well, although we won't be talking about these today. But things like food, dust, particles, sediments, soil, even personal monitoring devices like silicone bracelets are all sources of these chemicals that we can then look at using these bioanalytical tools. For water samples, these water samples can be collected either by grab samples, where you're collecting the water and then bringing them into the lab, or they could be through passive sampling procedures as well. The sample extraction is very similar to how you might do sample extraction and concentration if you were to do the traditional analytical chemistry approach. But in this case, the goal is to look at not only the knowns, but the unknowns, all these different contaminants of emerging concern. So we don't have a developed procedure, for example, like we do with extracting PCBs from different matrices. We don't have that set EPA guideline for how to extract. What we want to do is be able to get the widest variety of these contaminants that we can. This can be through either solid phase extraction procedures or liquid extraction procedures, but we're going to want to get a variety of both hydrophobic and hydrophilic compounds, which would represent the pharmaceuticals, these persistent pollutants, pesticides, flame retardants, plasticizers, etc. The next part of this, after we've determined how we're going to do the extraction and the concentration, is try to understand, well, what are the biological endpoints that we want to assess from a water quality standpoint. And with this variety of compounds, we could look at a variety of different toxic endpoints. Some of the most common endpoints that are of concern with these contaminants are endocrine disruption, cancer, inflammation. You may also be concerned with neurotoxic effects, etc. A lot of the recent studies using these bioassays have kind of focused in on a very important subset of contaminants, and those are the ones that are called endocrine disruptors, or I like to call endocrine and metabolic disruptors to be more inclusive for these types of chemicals. And endocrine disrupting compounds, they're found, they are defined in many cases as these contaminants of emerging concern. They can be household products, they could be from industry, they could be from agricultural activity, pesticides, etc. And they're basically just defined as chemicals that interfere with the synthesis, secretion, transport, binding, elimination of natural hormones. And of course, natural hormones are very important for normal cellular homeostasis, but also for important effects such as development, behavior, fertility, cancer, etc. So by looking at endocrine disruptors, we really kind of encompassed a lot of these contaminants of emerging concern, and also an endpoint that is of very much interest, not only to human health, but also to environmental health. The last step, now that we've kind of narrowed down the type of endpoint that we want to look at, focusing on these endocrine and metabolic disrupting chemicals, is now we have to match the bioassay so that we can interrogate that particular endpoint. And so this is where we're going to spend the rest of this talk. So what are bioassays? How do we choose which bioassay to run? And then how do we determine the source or the whose bioassay we're going to utilize? So first of all, what are bioassays? And bioassays are basically just simple systems that you can use in a laboratory to analyze different biological effects of compounds or of extracts, complex mixtures, but we're really looking at something within an organism, how it's affecting some component of the organism and not the organism as a whole. And these endpoints can vary. These can be at the cellular or molecular level, looking at genes, proteins, etc., or it can be looking at a higher biological response. So looking at a more coordinated response instead of just an examination of one particular endpoint. Now we're looking at how these endpoints are interacting with each other. Cell proliferation, cellular function types of assays fit into this bioassay. These are generally in vitro, but they don't need to be. And I'm using this term in vitro in the classical sense, talking about looking at the effect in a test tube. But as we'll see, these are actually live cell assays that are being used. So you could also use the word in cello to describe these particular bioassays. So these bioassays, again, are looking at these effects on a cellular basis. And we can pick which cell type that we look at that might be indicative of a particular response to a chemical. We can look at simple organism effects as well, would fit into this bioassay. For example, embryo development, or looking at zebrafish or some other model that has the advantage of being more high throughput. And again, what's the advantage of doing these bioassays as opposed to just straight analytical analysis is that we're putting some biological context to this. And if we're doing this correctly, not only are we putting a biological context to this, but we're putting a toxicological context to it as well. And of course, one of the benefits, in addition to this biological context, is that we're able to look at the cumulative effect of all of the different components that we've added to the cell in a mixture. We could also look at this with known chemicals as well as unknown chemicals and have a predictive type of response. And also an advantage of these bioassays as opposed to doing animal studies or some other preclinical study is these can be done in a very high throughput manner, short amount of time, low expense sort of studies. Well, let's talk about which bioassays are we going to choose to address the quality of a particular water sample. And I'll just put down what we must have in a bioassay and what would be nice to have. Must have, at least from my perspective, if we're going to try to make decisions on the quality of a particular water source, is we're going to want to have a bioassay that is predictive of a toxicological response, of a hazard. And so that we can make that link between the presence of an activity and the presence of a potential adverse event. We also want this bioassay to be responsive to known contaminants of emerging concern, things that we know that are in the environment. We want to be able to address the environmental samples and whether they've been looked at in environmental matrices in the past and correlated with the presence of contaminants of emerging concern. And is this something that we can routinely add into water quality studies? And this would include issues such as cost, how much technology and resources are necessary, what expertise is necessary to address this. So what we're looking for is low cost, easily addressable technology with easily accessible expertise. Some things that are nice to have that hopefully will be able to move some existing assays into the workflow is what we want to have is standardized bioassays where we've established that they work, with particular reference compounds of known toxicity and of known presence in the environment, and where we can compare one lab to another lab and come up with the same types of results. And something that I'll address later on is having bioassays that we are able to make decisions with, where we're able to say we have this amount of activity in our bioassay. That means that we have or have not reached a particular trigger value. That means that we have safe or unsafe water. So let's kind of walk through some of these must-haves, and we'll come back to the nice-to-haves later on. Okay, so one of the must-haves is to have something that is predictive of toxicity. And what we take advantage of here is efforts that have been put forward by the Environmental Protection Agency here in the US and other government agencies in the EU and in Asia that are trying to address the fact that we have so many chemicals that we do not have any toxicity data on. So what we want to do is develop predictive pathways that we can use as screening tools to look at unknowns. And these are called adverse outcome pathways. And these are pathways of known events that have been shown to be associated with an adverse event. So something that is predictive. If this event happens, it's followed by this event that leads to a toxic response. And the way that these are being framed, these adverse outcome pathways, are these linked pathways that have three major components. We have an initiating event that is linked to, so we have a relationship to other key events that, again, is predictive of some adverse outcome. So the first event that occurs in an adverse outcome pathway is a molecular initiating event. And so this is the chemical interacting with the organism, in particular a macromolecule within the target cell of that organism. And this interaction between the chemical and this molecular initiating event starts a series of events within that target cell. And these are called key events. So these can be altered signaling within the cell, changes in gene expression, changes in interactions between cells. These key events are linked to other key events that are altering the state or the fate of that particular cell, how it develops, whether it proliferates. And those events then are tied to the adverse event. So what we want to do is rely on these different pathways when we are trying to find our bioassays to use. So what are some of these adverse outcome pathways? And as I mentioned before, what we're predominantly interested in addressing with these bioassays are endocrine and metabolic disruption. So there have been clearly established adverse outcome pathways for endocrine and metabolic disruption, which are shown to the left of this slide. So the chemical interacts with these molecular initiating events for both endocrine disruption and metabolic disruption. These lead to altered cellular events, such as changes in gene expression, which result in altered cellular states. For endocrine disruption, we have interactions with receptors such as the androgen receptor, estrogen receptor, thyroid hormone receptor. For metabolic disruption, this is also called the adaptive response, where we're responding to these environmental chemicals by trying to increase their metabolism and excretion. And in this case, these compounds are interacting with a whole slew of different molecular initiating events, driving a variety of different drug and hormone metabolism enzymes, which then are going to affect a variety of different molecules within that organism, which could lead to drug-drug interactions as well as endocrine disruption. So as you can see from these adverse outcome pathways, there are a variety of molecular initiating events, and these all fit into a category which are called ligand-activated transcription factors. So they recognize a particular chemical, and then they drive gene expression in response to that chemical. And one of the overlying families of chemicals within these two adverse outcome pathways that are these ligand-activated transcription factors are called nuclear receptors. And these are important xenobiotic responsive transcription factors that respond to a wide variety of different drugs, pollutants, nutrients, and endogenous hormones. Things like the estrogen receptor, which responds to estrogens, androgen receptor, which responds to testosterone, thyroid hormone receptor, thyroid hormone, etc. So we are very interested in linking the environmental chemicals with these particular molecular initiating events because these are tied to that metabolic and endocrine disruption. The third category is for determining which bioassay to use is we want to make sure that they are that they're able to detect known contaminants of environmental concern. So in order to look at what are the known molecular initiating events, for example, for these contaminants, we can use some public databases such as the EPA CompTox dashboard. And what I'm showing over here is just a simple search looking at a EPA list of contaminant candidate list. In particular, this is called CCL4, which is 97 chemicals that are commercial chemicals, pesticides, toxins, disinfectant byproducts, et cetera, that we know are in a variety of different water sources here in the US. All of these compounds have been studied with the EPA ToxCast program, where they basically looked at a variety of different bioassays and asked what does this chemical interact with? And they provide that information in this ToxCast database. And so what I've done is simply looked at these 97 different compounds and asked what are the common bioassays that showed a positive. And so anything that you see over to the right means that they've been detected several times in this 97 chemical portfolio. Anything that moves along this y-axis means that there's a high percentage of these 97 compounds that interact with these particular targets. For example, over 40% of these compounds interact with the androgen receptor, estrogen receptor, progesterone receptor, pregnant X receptor, and CAR. The other thing that's important is if you look at some of the key contaminants of emerging concern, things like bisphenol A or PFOA, PFOS, the phthalates, that our bioassay should be able to detect those chemicals in the environment as well. So again, this is looking at BPA and the ToxCast program, looking at AC50 values, so potency, at the ability to regulate these different assays. Sorry about that. Anything to the left on this graph means that it's a high potency effect. Anything moving up the Y would be a high activity effect. And if we look over to the left side, which would be the high potency activation, again, we see some of the same players that we noticed up here. So things like estrogen receptor, PXR, CAR, but also some other compounds or receptors such as the PPARs. The next step in trying to pick our assays is have these been detected in environmental water samples in the past? So for this analysis, what I've done is I just went to PubMed and looked for papers that looked at a variety of different biological sources, looked for in vitro bioassay, bioanalytical tools, effect-based methods, cell-based bioassays, and simply collected all that data. From that search, there's been roughly 513 papers. You can see that a nice trend for an increase in interest in this area. It's a variety of different biological matrices that these papers come from, including wastewater and surface water. And now if we start to look individually at some of the targets from these manuscripts, again, here's the number of times that each of these different bioassays has been examined as you move to the right. And then how many times that it was examined did we have a positive above background levels detection. So we're looking for things that are above, let's say, the 50%. And again, we can see bioassays, molecular initiating events such as the estrogen receptor, androgen receptor, aryl hydrocarbon receptor, glucocorticoid receptor. All right, the final feature that we need to consider is now we're starting to refine which bioassays we should be looking at. Now let's address the question, can we look at them? Are we able to develop assays that are able to examine the presence of these types of compounds? So there's really two different approaches that you can use here. You can do the do-it-yourself assay, or you can use commercially available assays. And basically, do-it-yourself assays, you are taking a cell and you're putting in your molecular initiating event. And then you're putting in the machinery that's required to develop that key event, namely gene expression. But the key feature to developing your own homegrown assay to look at a molecular initiating event is to develop it so that it does have that high throughput capability as well as high quantifiability and sensitivity. So what most people do for these types of assays is they use what's called a reporter gene, where you are putting in a gene that is responsive to your molecular initiating event. And this would change in activity when you activate or interact with your molecular initiating event. Your reporter gene can be anything that's easily measurable. These could be something that's fluorescent or luminescent, but it has to be something that is able to be easily detected and has a high throughput format. Now, I went through that very quickly because there are many commercially available either services or products where you no longer have to use this do-it-yourself approach. You can simply ask a company to look at these, and they have these basically store-bought assay systems ready to go for a particular molecular initiating event. So there are several companies that are out there, both within the US and internationally. But Indigo Biosciences has the largest amount of the human nuclear receptors, as well as some of these other target factors, as well as some other non-human orthologs. In particular, of particular interest for the environmental area is zebrafish assays. And these come in a variety of different formats, 96, 384 well formats. I'll talk about the utility of these 3x32 a little bit later. So these are something that you can purchase if you do not have the capability to do the do-it-yourself. Just a little bit more information about why we've decided to use the cell-based assays. And really it's giving you more information per bioassay than you get from a biochemical assay. So you can look at increases as well as decreases in activity, so agonism as well as antagonism. You can look at full agonists versus partial agonists. You can look at cell viability at the same time as you're looking at these biological effects. They're easy to use, they're highly quantifiable. What you would need as far as resources and expertise, if you're farming this out, of course, all you really need is your samples. But if you're purchasing the kits to do in your own lab, all you really need is some mammalian cell culture facilities and expertise. And then you need the ability to detect the reporter gene, a luminometer or a fluorometer or a spectrophotometer, depending on what your reporter gene is. Okay, so from this discussion, we've kind of come up with some clear winners of what are bioassays that we can use to address water quality. Certainly, the estrogen receptor has been shown to interact with a variety of different compounds of emerging concern, clearly associated with endocrine disruption. The aryl hydrocarbon receptor interacts with a wide variety of, in particular, lipid-soluble ligands, and it drives that adaptive response. It regulates the metabolic disruption part of the endocrine and metabolic disruption. They've been highly examined, they've highly standardized assays, and several regulatory agencies have moved these two assays forward into some of their thought process of looking at water quality. And I'll show you some assays that we've developed in our labs looking at estrogen receptor as well as the aryl hydrocarbon receptor. But really what we want to do eventually is validate all of these other assays so that we can look at different endpoints, or certainly so that we can look at other endpoints that we know are important for contaminants of emerging concern. The other thing that this process at least solidified in my mind is that there are several of these receptors that are not being utilized in these environmental samples that should start to see the light of day. Things like PXR, CAR, and NERV2, which are involved not only in this xenobiotic metabolism or metabolic disruption, but also directly involved in things like inflammation. All right, I'm going to just kind of quickly go through some examples that we've generated in our labs and kind of go through some of the steps that you may have to go through in order to incorporate these types of assays into your thought and development process. The first is you have to get your assay developed, get it in hand, and start to determine several factors about your assay. How sensitive is this to your reference compounds? So over here I have estrogen receptor being activated by 17-beta estradiol, the aryl hydrocarbon receptor being regulated by dioxin. And so these are the reference compounds. You can start to look at your dose response, looking at your potency, the EC50 values, as well as what is your limit of detection. And once you determine in your hands what your limit of detection is, then you can start to assess this relative to published detection limits for other assays, what sorts of detection limits are required for different environmental samples, and then make sure that your assay system is sensitive, robust enough in order to evaluate the presence of these compounds in the environment. The next step would be to start looking at environmental samples. Generally speaking, all of these types of chemicals are present at low levels in these matrices, so you're going to have to concentrate them. So generally what we do is we'll take a liter of water, go through SPE extraction, resuspend in 100 microliters of a solvent that's compatible with cell-based systems, DMSO, get an enrichment factor of 10,000. And then when we go into the bioassay, we're using a dilution factor of 1,000. So we have 10 times the concentration of the chemicals in our media that we would have in the environment. And then we can make dilutions as far as that. So now when you're examining your environmental compounds, what you're doing is you're saying, I'm getting a certain amount of activity in this bioassay. How much is that relative to the reference compound? So what I'm showing over here is the amount of reporter activity that we're getting with water samples relative to estrogen receptor alpha. I'm sorry, to the aryl hydrocarbon receptor. So what we're looking at is how much activity in the sample expressed relative to dioxin. And so we end up getting what are called bioequivalent factors. So we are determining how much of activity we get relative to the reference compound. Now we can start to use this to make decisions. So we have these bioequivalents, and now we can start to to examine a variety of different samples. And remember that this is looking at the sum of all activity on that receptor. So for the aryl hydrocarbon receptor, we're not just looking at dioxin, we're looking at all chemicals that mimic dioxin, that have an activity that is similar to that, and are predicted to have the same toxicity that dioxin would have. And so shown to the right here, for example, are 25 different water samples that have gone through this extraction procedure. And now we can look at the amount of activity towards this receptor and dioxin equivalents, and start to compare which sites have the highest activity, have the highest potential for toxicity. We can also look at this approach to see what is the effect of treatment of a particular water sample. So what's shown here is a wastewater treatment sample before and after going through a detoxification process, treating it with manganese oxide to break down some of these bioactive molecules. And you can clearly see that we were very effective at degrading that bioactivity. We have rendered this less harmful by our treatment. So it's very useful to get this kind of quality assessment, qualitative assessment, before and after treatment, as well as comparing and prioritizing sites. You can also do a sanity check. So you're looking at the presence of all the different chemicals using analytical approaches, and then you see if this matches what you're seeing for aryl hydrocarbon receptor activity. Are you seeing a lot of estrogen-like compounds in the pre-treatment that we have degraded in our post-treatment? So you can start to make sure that your bioassays are complementing what you're seeing with your chemical analytical tools. I'm going to quickly kind of go through another example that is kind of taking this to the next step. So those individual assays are very useful for looking at these very well-defined endpoints, the estrogen receptor and the aryl hydrocarbon receptor. But one of our interests is to be able to look at more endpoints and be able to look at more complex mixtures that may have different types of biological activities. So what I'm going to describe next are taking, instead of an individual receptor, taking a panel or multiplexing several assays together to kind of look at a biological niche or a grouping of different receptors that all have the same activity. So what we're doing here is taking individual cell-based assays, mixing them together, pooling them, and then dispensing these into a 96-well plate, then adding our extracts. And we're asking the question, are our extracts activating any of the receptors within this pool? Then, if we are getting an activation, then we can go to the next step and say, well, which receptor is this activity coming from? And in this way, we can do a smaller set of initial studies to get the landscape of all the different types of activities, and then we can come back and look at the individual receptors, as opposed to looking at each individual receptor from the beginning. So here's an example of some of the experiments that we've done. In this case, we've pooled basically 30-some receptors and put them into groups of 5 or 6. Put them into an individual pool, and each of these groups of 5 or 6 have a particular biological pathway that we're looking at. So our metabolic disruption, lipid metabolism, our reproductive and development, our endocrine disruption, basal metabolic functions, and then inflammation and tox. And if you do this initial screening with some important contaminants of concern, let's look at the triphenyltin over here on the right. You can see in the panel approach, we're getting regulation of this lipid metabolism, as well as the basal metabolic functions, take a look at which individual receptors are responsible, and you can see that we're getting activation of the PPARs, we're getting activation of the RXRs. So we didn't have to look at each individual receptors. We looked at the pool, and then we were able to determine which particular receptors to look at next. And this approach can be used for complex mixtures as well. And without going into a lot of detail here, we can start to kind of get a overview of what is happening. And so, for example, in the wastewater treatment, the same example that I showed you before, but now using a panel approach, we can see that our two different wastewater treatment samples, high level of endocrine disruption activity, some basal and metabolic disruption activity with wastewater treatment 2. You can look to see which receptors it's coming from. It's coming from estrogen receptor in the case of wastewater treatment facility 2, and androgen, estrogen, and RXR, or RAR, I'm sorry, with the wastewater treatment facility 2. One of the reasons why I like to show this particular kind of big picture is now you can start to look at water samples from different sources, and you can see how different they are. So for example, compare this wastewater treatment facility sample here on the left, what its biological activity is, and compare it to sample 3, which is a sediment sample, which was extracted. And you can see that in this case, we're getting more hydrophobic compounds that are able to regulate the metabolic disruption. So you can get a kind of a different picture of the types of responses that from different water samples. Where do we want to go with this? I've already mentioned that there are a couple of these assays that are moved along quicker, that are close to getting to what are called effect-based trigger values, where if you know you have a certain amount of activity, then this means that you have reached a threshold where you have a water sample that is unsafe or is has potential hazardous effects if this gets into the environment or into our drinking water. So one of the difficulties with taking these effect-based pathways is that these are mixtures. These have multiple effects that are cumulative, plus there are many unknown compounds. So we're trying to get a better grip of how to use some of our standard approaches that we use for looking at water safety, but now incorporating in these effect-based. So I think this is an area where we really need to do some more work, some more thinking. We need to go through it and look at reference compounds of known activity where we do have these guideline values, and then try to come up with ways of looking at these more complex mixtures. So this is something that I know a lot of agencies are interested in moving toward, but it's going to take a lot more work for us to have comfort with that. All right, to wrap things up, and I know I've gone a little long here, but I hope that you appreciate at least that these effect and mechanism-based assays have a lot of utility, and they are a great complement to the traditional analytical chemistry approach. So they're not, at least in my view, I don't think they're ready to completely relieve the analytical chemistry approach, but at least they are adding to it and adding functional information. So they're adding that biological and toxicological context, in addition to the fact that these are looking at cumulative effects of multiple chemicals acting through the same mechanism, as well as looking at agonism, antagonism, and the effects of the interaction of chemicals within this mixture. Amenable to high throughput screening, relatively easy to perform and interpret. There are many different bioassays that are available, both for service assays as well as for products, but we still have some work to do. We still have to be able to standardize the assays for use in multiple labs. We have to come up with what are the reference compounds, what are the best ways to quantify And then, of course, getting to that last point that I mentioned, how do we make clear biologically relevant and toxicologically relevant decisions based on this data? So with that, I'd like to thank the people who were collaborators at Penn State as well as at Indigo. In particular, I'd like to thank Megan Granda within my lab, who did all of the different reporter assays that I showed. And then, of course, the team at Indigo, including Bruce Scherf, Kayla Smith, Dan McNulty, who helped put this webinar together, and of course, our CEO, Fred Moroney. And with that, I'd be happy to take any questions.
Well, thank you very much, Jack, for that insightful presentation. And that does bring us to our audience Q&A. So audience members, you can still continue sending in questions to Jack. We've already received some questions, so I'll start off with those. And our first question, Jack, is, are there recommendations for extraction procedures that are compatible with bioassays?
Yeah, there's been quite a bit of work with that. And there are some procedures that have kind of moved forward and are used by most labs. And as I mentioned, you need procedures that are able to get a wide variety of different chemical structures. So SPE is the easiest procedure to use. It's amenable to not only high throughput, but requires a low level of expertise to initiate. And the key thing there is to pick the right matrix to do the extractions. And so you want something that will interact with hydrophobic as well as hydrophilic compounds. And so there are different matrices. The one that has been most used is called HLB columns, like Oasis HLB. Some people use C18 columns. So there isn't a standard approach, but we're getting close to having some of these SPE procedures.
Great, thank you, Jack. And our next question is, what expertise is required to run these assays in your own lab?
Yeah, I touched on this a little bit, but basically, if you're going to run these in your own lab, all you need is cell biology experience. So you need things like a biosafety cabinet for doing sterile work. You would need an incubator, a water jacketed incubator at 37 degrees with CO2. You would need the expertise to do aseptic technique, and then just simple biochemical assay tools like being able to look at luminescence or fluorescence as your endpoint. And incidentally, I mean, I've trained undergraduate students to do these assays. I've even had high school students run these assays. I've even run them myself. So that means that you don't have to have a lot of expertise.
Fantastic. And another interesting question from an audience member, and that question is, activation of the estrogen receptor and potential for endocrine disruption is easy to understand. You mentioned that AHR is often used to examine water quality. How do you interpret activation of this receptor to health and safety?
Yeah, that's a difficult question. And I totally agree that activation of the estrogen receptor, you can appreciate how that is affecting the endocrine system, because estradiol, what is the natural ligand for the estrogen receptor, is the hormone that causes that's involved in reproduction and development. And so interfering with that, I think, has a clear biological context. The aryl hydrocarbon receptor is a less well understood receptor. We don't know what the natural ligand is. All we know is what the effects are of activating that particular pathway. And we know it drives a variety of different enzymes and proteins that have been associated with metabolizing different hormones. So one of the things that we know is that from a hormone standpoint is if you activate aryl hydrocarbon receptor with things like dioxin, this interferes with thyroid hormone activity by increasing the metabolism and breakdown of thyroid hormone. So it's a little harder. It's like one step removed from the estrogen receptor, but it is still affecting how hormones work and affecting the activity levels of different endogenous signaling molecules.
Great, thank you so much, Jack, and thank you for all those questions, audience members. We've reached the end of the Q&A portion of this webinar. If you couldn't attend to your questions, the team at Indigo Biosciences may follow up with you later on as well, so not to worry. And if you do have further questions, you can also direct them to Jack, and there you go. So you can see his email address there. That's jackvh@indigobiosciences.com. So you can go ahead and quickly jot that down. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us improve our webinars. Now, I'm about to send you a link in your chat box. You'll be able to view the recording of this event on this page, and you can also share this with your colleagues if they want to also watch that recording. So go ahead and take a little note of that link that I just sent in the chat box as well. And now please join us in giving a big thank you to our speaker, Dr. Jack Vanden Heuvel, for that fantastic presentation. We hope you found this webinar informative. Have a great day, everyone.